Welcome to the Wednesday afternoon lecture series. I am Susan Buchanan. I am the Deputy Scientific Director for the National Institute of Diabetes and Digestive and Kidney Diseases. And I'm also the Chief of the Laboratory of Molecular Biology, where our speaker hails from. So it's a real pleasure for me to be giving this introduction. And I'm excited today to introduce our speaker, Dr. Eric Greene, who will be discussing single molecule studies of homologous recombination. That's Eric C. Greene with a final E, not to be confused with our NHGRI director, Dr. Eric D. Greene, without that final E. But anyway, I think you figured that out. So our speaker for today is Eric Greene, who is a professor at in the Department of Biochemistry and Molecular Biophysics at Columbia University in New York City. His lab uses single molecule optical microscopy to study fundamental interactions between proteins and nucleic acids. In essence, to observe individual protein molecules or protein complexes as they interact with DNA substrates. Indeed, Eric's lab, excuse me, has pioneered the development of DNA curtains for this direct visualization of hundreds to thousands of individual molecules in real time by optical microscopy. The technique is based on the use of nanofabricated surfaces in combination with lipid bilayers to arrange DNA molecules in defined patterns, allowing them to be viewed at the single molecule level by total internal reflection fluorescence microscopy. Now, many of you may remember Eric was a postdoc here in the Laboratory of Molecular Biology with Kiyoshi Mizuchi at NIDDK from roughly 1999 to 2003. I was here then too, and that's hard to believe. It's been 20 years plus. Eric received his undergraduate degree in biochemistry from the University of Illinois, Champaign-Urbana. He followed that with a PhD in biochemistry from Texas A&M. Then he did his postdoc at the NIH, as I've told you. And after leaving the NIH, he took a position at Columbia University in 2004 as an assistant professor, where he's been ever since, being promoted to associate professor in 2010 and full professor in 2015. Excuse me. He has had many honors. Some of them are an NSF Career Award in 2006, a Presidential Engineering, and I forgot what it stands for, PCASE Award in 2007. I should know, I had one too. A Howard Hughes Medical Institute Award from 2009 to 15, the Dr. Harold and Golden Lamport Research Award in Basic Sciences in 2009, and multiple invitations to be a keynote speaker, notably recently at Gordon Conference and a Keystone Symposium. He gives an engaging lecture, and the title of his talk is Single Molecule Studies of Homologous Recombination. Eric, welcome back to the NIH. We wish you were here in person, but thank you very much for being with us today.
Perfect. Thank you, Susan. I also wish that I was there. I had sort of agreed to do this, assuming that I'd be able to come down and visit with so many friends. So that's a little bit of a bummer. It's also funny that you brought up the fact that there's another Eric Greene at NIH, and it reminded me of a story when I was a postdoc and some lawyer called me and started talking about some things that obviously I had no need to know. And I realized that I was the wrong Eric Greene. So that was fun. Today, I was kind of thinking about what it is I should talk to you about. And I decided to talk to you about something that's not published. So the talk's a little rough. I was originally going to say something about our recent work on homology search, but I decided to talk about how DNA ends are processed, or at least our small portion of the puzzle in terms of DNA end processing. And you can see I've changed the title to something a little more spiffy. I'm talking about Bloom Helicase. And what I'm going to try to tell you about is how Bloom Helicase resects DNA ends. And I'm going to try to convince you that it's doing something to those ends that looks like it's making some kind of a loop with the single-stranded overhang that's being produced. And so with that, I will jump right in. And I'm going to point out the folks who have contributed to this. Really, from my laboratory, Chaoyu Zhu, who's now a former postdoc and has his own lab back in China, will have done all of the single molecule work that I'm going to talk to you about. And we've been working closely with Patrick Sung at UT San Antonio, and I should say Samir Salunki has been sort of the main driver of the work in his lab. So anytime I'm showing you something that is a live cell assay, that's Samir. That has nothing to do with us. And then I should also mention Sandeep Burma and Nozomi Tomomatsu, who came in at the end and helped us with some stuff that we also had to get done in terms of making measurements. And I'll try to point out who among these folks have done what as we've gone along. And of course, I have to say thank you to all of our funding, most of which is NIH. So this is Chaoyu, sort of midst of a pandemic as he was preparing his family to move back to China. And he published a number of papers with us, and some of these I'll be referring back to from time to time. So if anyone wants to see the full references, they're going to be listed in this slide. The key ones here are going to be a study that he did on end resection in Saccharomyces cerevisiae. That's this PNAS paper and a molecular cell paper, as well as a study he did on human bloom helicase, which I'll talk about in just a minute, that we published in Nucleic Acids Research. So this is Chaoyu, and man, he was great. I really miss him. And so I'm going to keep the introduction to this fairly simple. I really only have 2 introductory slides, and then we're going to delve right into data. And then at the end, I'll try to tell you what we think the data means if I haven't convinced you as we go along. And so over the years, my lab has worked on lots of stuff. And I'll show you the tool in the next slide that we use to do this, but we've been coming more and more towards focusing on homologous DNA recombination. It's interesting because when I first started my lab, this is what we were going to be working on. And you know, as we developed techniques and realized we could apply these to lots of systems, that's exactly what we did. But we've had some breakthroughs that allowed us to come back to recombination in ways that I think have been very exciting for the group. And so this is sort of the biophysics overview of DNA recombination. My genetics colleagues love this, right? So we have a double-strand DNA molecule, a little lightning bolt comes down and somehow damages it. That could be all kinds of things that will do this to generate a double-stranded break. And for homologous recombination, that break has to be processed. And in fact, the processing is what we're going to be talking about today. And so what happens is you generate a long 3' overhang. And this overhang serves as the platform for the formation of what's called a presynaptic complex. And that's going to be in humans, a filament of a protein called Rad51. Okay, this same process happens in even bacteria. The difference is Rad51 is not there. Instead, you have a protein called Rec-A. And it turns out that Rec-A and Rad51 are very, very closely related proteins. And so the process in and of itself is very, very highly conserved across biology. Now, once you've resected these ends and you assemble your recombinase filament, that filament then has to search the genome to find another piece of DNA, here depicted as a red molecule, that it can pair the black DNA with. And once that pairing takes place, I can get my cursor back, you can then start to do DNA synthesis and fill in information that was lost during these stages. And then this intermediate can be channeled through a number of interrelated pathways, the most simple of which is to have this intermediate disrupted. This newly synthesized strand simply pairs then with this second overhang, and you get reannealing or religation of the strands after some further processing. That's the simplest, but there are other ways to do this as well. I'm not going to go over those because we're not going to be talking about them in depth. Okay, so now recombination in and of itself is important for maintaining genome integrity in essentially every organism. It's intimately involved in the repair of DNA damage and, of course, in cancer biology. And so I'm not really sure what the makeup of the audience is right now, but many of you will have heard of the proteins BRCA1 and BRCA2, these sort of cancer-associated oncogenes. And these guys, when they're mutated, you have a high propensity to have, for example, hereditary breast or ovarian cancers. And what you're doing there is actually messing up this portion of the reaction where you're trying to assemble a filament. And so there's an intimate relationship between cancer biology and homologous recombination. And in fact, mutations in proteins that happen along this pathway are often associated with cancer-prone syndromes. In fact, one of the proteins we're going to talk about today is just like this. And of course, defects can lead to chromosomal rearrangements, changes in copy number, lots of bad things. And I should note also that recombination is intimately involved in the rescue of stalled or damaged or collapsed replication forks. And so this is it's really essential for that. It's also involved in horizontal gene transfer. So if you're doing some sort of CRISPR type of thing, chances are you're relying on recombination to insert a gene somewhere. And then, of course, for the folks that are interested in meiosis, this is the process by which genetic diversity is established in meiosis. It's through recombination between different DNAs. Now, we have studied reactions along this pathway in a number of different reactions, a lot of which have focused on the assembly of these nucleoprotein filaments on these processed DNA ends, as well as the pairing processes that take place to get your black DNA paired with your red DNA. But today, what I'm going to talk about is how you go from this double-strand break to a processed double-strand break where you've resected these ends back. And I should point out that I'm going to talk about three proteins, Bloom helicase, DNA2, and RPA. I'll tell you what these are in just a moment, with the understanding that we're using a very simplified system to try to look at what we think are sort of key aspects of the reaction. And in reality, inside of cells, this is going to involve a larger number of proteins that we hope to eventually be able to look at in our reconstituted system. But for the time being, we're looking at a minimal system. Okay. So now, Bloom helicase, the first of these guys, and again, you'll hear these three names throughout, is a RET-Q helicase. And when you have defects in it, you actually end up with a cancer-prone syndrome, Bloom syndrome, that's characterized by really profound DNA repair defects and chromosome rearrangements. And so the folks, the patients that have this end up having cancer at a very, very young age, unfortunately. Now, the second protein that I'm going to talk about is DNA2. It is also a helicase. It's not quite as robust as BLM, but it's also a nuclease. And so DNA2 here is actually what's responsible for cutting out this other strand of DNA to leave a 3' overhang. And then RPA is replication protein A. It's a heterotrimeric single-stranded binding protein. And so this is a protein that's essentially involved in anything involving single-stranded DNA inside of a eukaryotic cell. So it's a highly abundant protein and it's ubiquitous. It's involved in everything. And so these three players are what we're going to talk about in terms of generating this processed DNA end. Okay. So now, just so everybody's on board with the technology that we'll be talking about, this is what we call DNA curtains. Essentially, we have a flow cell that we make where we have an inlet and outlet chamber, a little sample chamber in between that's made with very fancy double-sided sticky tape, if you can believe that. And then what's really special about what we're doing is we're modifying the surface of this chamber. And exactly what we're doing is putting down patterns of metal or metallic barriers. And in this case, what I'm showing you is a blow-up a schematized blow-up of a zigzag-shaped pattern. It's made out of chrome that we deposit onto that surface by e-beam lithography. And I'm showing you one, but we actually put these all over the place in here. And what we then do is take this surface, this glass surface, and we cover it with a fluid lipid bilayer, okay? Now, the bilayer is serving two purposes. The first is that it's going to ensure that this surface is as inert as possible in terms of us putting proteins into these chambers. And so proteins love to stick to glass. If you were to just inject any protein that we would work with into a glass chamber, a few silica chamber, it would simply coat the surface, and that would be the end of your experiment. You wouldn't be able to tell anything. And so the bilayer is our effort to try to make this chamber surface look more like the inside of a living cell than glass, okay? Now, the other purpose that it serves is a subset of the lipids within the bilayer have a biotin group, and we then couple DNA molecules to that surface through a biotin-streptavidin interaction. Now, the biotin or the lipid bilayer itself is fluid in 2 dimensions, and that means when we attach things to it, they can diffuse around, attach to the surface. And what we do then is apply a flow force or hydrodynamic force that pushes the DNA into this barrier. Okay, so the DNA is acting essentially like a sail on a ship. It's pushing them into the barrier. They then lay down parallel to the surface, and then we're illuminating them with total internal reflection fluorescence microscopy. So we're essentially illuminating a very small excitation volume near the surface within the volume where these DNA molecules reside. Okay, and we've made many flavors of these curtains, many different styles. The ones I'm going to be talking about today are as shown here, meaning one end of the DNA is attached and one end is free. And so this is what we would call a single end tethered DNA curtain. And just for anyone that wants any additional information, we've published many methods papers describing this work. And we've published a number of papers describing studies that have come out of the use of these types of things. Okay, so that's it for the introduction. I do want to make one more point now that I'm thinking about it. So in the subsequent slides, I'm going to be showing you chymographs. And what the chymographs are single, so the data we collect is videos of these molecules as they're interacting. And the chymographs are single 1-pixel slices through individual DNA molecules that are then projected through time. And I'll try to explain that more carefully in the first chymograph so that we're all on board with understanding how the experiment works. Okay, so now this is an example showing a schematic, showing the substrate that we're going to be working with. And so this is, again, our DNA curtain. Now focusing on the end of the molecule, what we've done is engineered that so that we have a 30 nucleotide single-stranded DNA overhang. And the intention here is to mimic a partially processed intermediate. So I mentioned earlier that there are a number of proteins that come into these reactions that we're not looking at. One of those is called the MRN complex, MRE11 Rad50 NBS1. And what it does is generates a little single-stranded overhang. And then what happens on that overhang is we can then load our reactions containing BLM, DNA2, and RPA. So this is sort of giving us a jump start on that processing reaction so that we can look at the downstream steps. And I'll point out we've published similar work with the yeast resection system using these same substrates. So now, this is a wide field image of a DNA curtain. The DNA is not labeled, so you do not see it. And what you do see is this B and this E. So the B is the barrier. That's where we've aligned our DNA molecules. And the E is the end of the DNA, so the free end of the molecule. And what we're looking at then is Bloom helicase bound to that end. And so Bloom is labeled in this case with GFP. We have buffer flowing from top to bottom. And I'm sure you can appreciate just from the image that you can see these green spots, and most of them seem to be co-aligned with what we say is the end of the DNA. And this is just taking a single image. This is now looking at that data sort of in aggregate, where N is 176 DNA molecules, and the bars represent the locations of the Bloom, the GFP Bloom complex. And so in most of the reactions that we do, with some exceptions, and I'll show you these later, the Bloom is accumulating at the DNA ends. And of course, we do see some bound internally, and I'm going to mention they behave differently. I'll talk about that in a little bit. So most of the proteins go to the end. They tend to stay there. So once they're bound, they have half-lives that exceed our experimental measurements under all the conditions that we've tested. And I note here that they're resistant to this protein RPA, and I make that note because when we did a similar study with the yeast system and put the protein SGS1 here, as soon as we inject RPA, it actually kicks SGS1 off. So it's an interesting difference between cerevisiae and humans that we don't fully understand yet. But we do know now that Bloom goes to the DNA ends, okay? So now this is our first example of a chymograph. And again, what I'm showing you, just to jump back, is going to be a single 1 pixel slice through one of these DNAs and showing the end-bound complex and then projected over time. And so what you can see is that that molecule, or that green spot, starts to move up. So it's moving up against the direction of buffer flow from E to now the B direction. And I'm going to point out there's little fluctuations in this molecule. Essentially what is happening here is that DNA is free. We don't have a tremendous amount of hydrodynamic force exerted on it. So it's sort of flipping back and forth a little bit in the buffer. Those aren't actual things the enzyme's doing. It's something that the DNA is doing more on a macroscopic scale. And so we can make measurements based on, here's where it started, here's what it ended. We know the time frame. This is a scale. And we can make measurements of how far did it go and how fast did it go. And I'm just going to point out, we did a control here where we injected yo-yo-1 at the end of the reaction. And yo-yo-1 is an intercalating dye that lets us see the DNA. And this first of all, confirms that the DNA is there. And second of all, it confirms that the DNA is actually shorter than it was when it started. So we know that resection is taking place. We also know that this resection is dependent on DNA2. And remember, DNA2 is the nuclease involved in this reaction. And we actually see no movement or no resection if we use a Bloom mutant where the active site involved in ATP binding, the active site is mutated from a lysine to an alanine. And so I just want to make point out a couple of details. So we can measure velocity, we can measure distance. So how fast does it go? How far does it go? In the absence of RPA, we're going about 10 base pairs per second and over distances of about 7 kb. It's not tremendously fast. We work with other motor proteins, other helicases that really do go fast, 1,000 base pairs per second. So this guy is pretty slow. It's not unexpected. The addition of RPA actually stimulates both the velocity and the processivity, but on the order of 50%. And I'm going to point out, I mentioned earlier that roughly 60 to 70% of the proteins are bound at the end, others are bound at internal sites. The internal bound BLM complexes are actually much faster. They're moving at about 100 base pairs per second. So something about having Bloom at the end of the DNA is actually causing it to go much slower. Okay, and I want to comment briefly on what fraction of proteins are actually showing this type of movement behavior. So if we only have BLM in the reaction, it's about 7.4%. Okay, as soon as we add DNA2 or RPA, its partner proteins, we go from, on the order of 7% up to 25%. So we get a big jump in the effectiveness of the reaction, or the fraction of active proteins, just by adding its partners. And if we add both of those guys together, that's when we actually get the most robust activity, when we're starting to approach 40% active. And I'm going to point out, there was a paper in Molecular Cell by a guy named Michael Sonian, who works for Ilya Finkelstein, who was a former postdoc. And they see very, very similar behaviors in terms of BLM and its response to the addition of partner proteins. Okay, so now what we wanted to know is, what is the status of the single-stranded DNA that this thing should be producing? So again, because we're looking at GFP BLM, we're not actually looking at the DNA, and we can't see what's going on. And so to try to do this, what we did was take RPA, the single-stranded binding protein, and fused it to mCherry so that we could watch it binding to the DNA end as it was produced. And in these reactions, what we tend to see is at the beginning, we don't get RPA binding because there isn't much single-stranded DNA there. And then over time, that RPA mCherry signal starts to increase, and we can show that 100% of the molecules that are moving show an increase in RPA binding that scales proportionally with time and distance. Okay. Now, when we first started doing this, we noted something quite interesting, and that is, well, we thought we were going to be making a 3' overhang, and that overhang should be extended. But what we see is the RPA keeps building and building, but instead of showing an extended end, it seems to be tracking very closely in space with the GFP BLM. And that's shown here in this merged image where you can see the white color coming from the bloom and the magenta sort of tracking very, very closely with it. And we see this in 100% of the cases that we look at. And in fact, we also saw this with the yeast system, and I think that Michael saw this, Soniat, in his molecular cell paper as well. Now, the difference is, in this system, what we also saw was occasionally these things would all of a sudden, in a single step, get much, much longer, okay? And that happened in about 30% of the cases. And so when we see something like this, where a DNA molecule goes from a shortened length to something that's much, much longer than it was just in the frame prior, that says to us that somehow this DNA was compacted into a structure that then opened up very, very quickly to lead to this longer overhang. And we know that this is single-stranded by virtue of the binding of single-stranded binding specific RPA, okay? And so what this told us was that maybe this DNA is forming a loop that is somehow tracking with GFP bloom as the process is going on, okay? So now, what we wanted to ask next was, well, what is going on with the actual ends of the DNA themselves? And so what Chow Yu did was make a substrate where he put a single ATO-565 dye either on the 3' end of our initial substrate or on the 5' end. And he showed then that BLM binds to these ends. You can see it here. You can see it in the distribution histograms. And we can see the ends of the DNA themselves now, rather than relying upon RPA to sort of see where things are going. Okay, so we know now that this system works. And what we did was then start tracking the ends during the processing reaction. And so this is a schematic of the reaction as we anticipated it might be taking place. So this is our substrate, GFP BLM, our DNA 2 molecule, and RPA. And we thought, okay, maybe it's going to do something like this where the 5' end is cleaved. This is exactly what we anticipated. The 5' end should get cut off. Or, you know, in principle, if it wasn't getting cleaved, maybe both ends just tracked with the complex and something funny was going on in terms of both of them forming a loop. Okay, and I should emphasize again, in this diagram, this is what we would think of as this DNA loop that might be forming. Okay, so this is what happened in about 87.5% of the cases. So this is GFP BLM moving. This faint signal is now our 5' ATO-565. And right here, right when you get ready to start moving, it gets cut off and just simply disappears from view. And so this is consistent with what we thought might be taking place. Now I'm going to point out that in about 12.5% of the cases, we see something different. And that is the GFP BLM starts moving and the 5' dye also starts tracking with it. And so this type of observation is consistent with both ends of the molecule being bound somehow and forming a loop as this thing is progressing along the DNA. Now, one thing that we cannot rule out in this, and in fact, this is what I think is probably happening, I suspect that when we see the 5' end tracking with the resection machinery, we're probably missing the DNA2 enzyme from these. So I'm assuming that it's not there, and that's why the DNA is not getting cut. I'm assuming that, but I don't know it yet. What we really wanted to do is make a GFP tag DNA2 and start tracking that molecule as well. Okay, so the vast majority of the cases, this is what we see. 5' end is cut off. 3' end, we assume now, is the thing making the loop. Okay, now this is to test that even further. So in this case, we're labeling the 3' end with our single dye. And this is what we thought, might take place. If the 3' end is tracking, we should see it following the resection machinery, which we are tracking by virtue of this GFP BLM. And if the loop is released, then we should see that dye suddenly move backwards. And so I'll point out, in 100% of our observations, that 3' end does track with the resection machinery. So the magenta dye here, the ATO565, co-localizes with the GFP, and the two of them move together along the DNA molecule as they're going. Now, this is different than what I've showed you before, and I want to point this out. So in this experiment, 99% of them showed no evidence of sudden release. Okay, this is the one that did. And so we see that the dye goes from this position all of a sudden down to this position, and that movement actually coincided with BLM, the motion of BLM stopping. We don't know if that's coincident or not. Now, in the previous examples, I said something like 30% did this. Here, we're only seeing 1%, or less than 1%, I should say. And I'm going to say the difference between this experiment and what I showed you before is that these reactions are intentionally done with unlabeled wild type RPA. That's why we can see the end of the DNA. If we try to label RPA, it signals too bright. We can't see the DNA itself. And so I suspect that the release of the loop that we are seeing in reactions with fluorescent RPA might be caused by having a GFP or an M cherry on the RPA itself. And so it's sort of a fortuitous discovery that was made in terms of this looping, just because there's some partial defect with having a GFP tag on the RPA. And if we don't have it, most of them don't get released. Okay. All right. So we think it's looping. We think that our data are most consistent with a model where the 5' gets cut off, the 3' end tracks then with the resection machinery as it moves along. So now we want to ask, what protein component might be involved in this? Is it BLM or might it be DNA2? And so what we did to test this, or what Chao Yu did, was simply perform the same reactions in the absence of DNA2, okay? And so this is an example showing no 3' end release. This is an example showing some 3' end release. And so what you can see is that with just BLM and RPA, the RPA co-localizes with the BLM as it's moving along, processing the DNA end. And in this case, you do get an example of sudden release. Now, again, 100% of the substrates that we look at show evidence for this type of loop formation. 42.5% in this case remain intact. The remainder show evidence of this sudden release, okay? And I should point out, these also, when we omit DNA2, the reactions are going faster in this case than they were before. And again, we think DNA2 is actually starting to slow things down a bit as the reactions are going. All right, so we think it loops. We think Bloom is primarily responsible for that. And so we wanted to try to investigate what part of Bloom might be involved. And so this is sort of my simplified diagram of what Bloom looks like. And that is, we have an ATPase helicase core. We have an extended N-terminal domain, and I have it listed as a oligomerization. It's also involved in a number of protein-protein interactions, including interactions with RPA, interactions with Rad51, and so on and so forth. We also have a RETQ C-terminal domain, a nuclear localization signal, and a helicase RNase A-like domain at the C-terminal. I think that's what it's called. And so what Chao Yu did was initially focus on the N-terminal domain, and he made a bunch of truncations where he either cut the whole thing off or he cut off sort of marching segments going from one end to the other. And what we're going to ask is, how do these proteins behave in terms of DNA end recognition? How do they behave in terms of movement, distance, velocity, and loop formation? Okay, and I'm just going to tell you that all of these guys were very, very similar to full-length BLM, except for one exception, and you'll see what that is in just a moment. So here's our diagram again, just so we're all oriented. As we're making these truncations, every single protein with an N-terminal truncation was still very efficiently targeted to the DNA ends in our assays. And so there doesn't seem to be any defect in DNA binding or DNA end recognition. We also looked at velocity, and I'm going to point out these are velocities measured in reactions with only BLM, and it's BLM just bound wherever it happened to might be. I think the way the reaction was done in this case, we didn't actually have free DNA ends, we had both ends tethered. But that's neither here nor there. The most important part is that they're all going roughly the same speed, except this N-terminal deletion where we cut the entire thing off. And I have this as 614, it should be 641, but it's the entire N-terminal deletion. Actually, it's going faster than the wild type full-length bloom, and it's going a heck of a lot farther than the full-length bloom as well, whereas the others all seem to go roughly the same distance. And so something about chopping that whole N-terminal domain off is making us go a little faster and a little farther as we're marching along the DNA molecule. So now, this is something else that Chow, you kind of noticed right away. And that is, if you can picture in your mind what those GFP foci that I showed in some of the early slides look like, we have no problem detecting that signal. It's bright. It's much brighter than one would expect if we were simply looking at a single GFP just based on our own experiences. But now when he's looking at this N-terminal deletion construct, all of a sudden it's much dimmer than all of the other guys that we've looked at, including full-length bloom and any of the other truncations. And so there's this sort of large decrease, about 80% decrease in terms of integrated signal intensity when we have that mutant. So even though it's going faster and farther, it looks like there's less at the end. And Chow, you did a little bit of a fancier measurement where he bombarded these end-bound proteins with laser light and asked, okay, how many photo-bleaching steps do we see? And this is an example with full-length BLM, GFP tag BLM, and we're seeing 1, 2, 3, 4, 5, 1, 2, 3, 4 steps. And that indicates to us that there should be 4 molecules of GFP bound. And if we look at, that's one example, if we look at a population distribution, that's more or less what we're seeing on the order of 3.6 plus or minus, 0.5. And I'm gonna point out, we also see what we think are probably aggregates where we can't actually measure the steps. It's just a lot of protein. We're just categorizing these as greater than 6. Now, when we look at our N-terminal deletion, and again, that's a typo, it should be 641. Primarily what we're seeing are single-step photobleaching events, where what it means is the protein is there, and then boom, it all of a sudden disappears. And that is most consistent with the idea that we have a single GFP bound there, meaning a single complex of GFP, a single GFP BLM protein. And then there are, of course, examples where we have a few more, but not very many. So we think somehow chopping off that N-terminal domain has made us go faster, it's made us go farther, and it's reduced our oligomeric state. Okay, now, here's one of the things that we found was interesting, and that is, this is showing BLM where the DNA gets looped, and by looped, I mean the 3' end in this case is tracking with the resection machinery, which we've demarked with RPA GFP in this case. And you'll see why in just a minute. This is when we cut off that N-terminal domain. So instead of seeing this co-localization through time, what we see is that that 3' end stays put over time. It actually starts to get a little bit longer out here. And you start to generate this long track of RPA. And so what we think is happening is at the leading edge of this RPA, that's where our GFP N-terminal deletion is going to be sitting. BLM is going to be sitting, moving, dragging DNA2 with it, chopping up one of the DNA strands, but the 3' end is unaffected. It's not generating a loop. And so we get this long extended structure of single-stranded DNA lit up by GFP RPA. And again, I'll point out, most of our truncations look comparable to full-length BLM in terms of the percent that undergo looping. And it's this one mutant where we've chopped off the entire N-terminal domain that seems to go from a looping proficient enzyme now to something that is no longer generating DNA loops. Okay, so we found that exciting. Now, we wanted to ask, Chow, you wanted to ask, what about the N-terminal or the C-terminal domain? And so he went about generating a number of different constructs here, where he either chopped off the entire C-terminal domain up to the RETQ-like C-terminal domain, including the HDRC and the nuclear localization signal in this dark blue and yellow regions, which we didn't really know what they were doing, although we know that they're involved in some protein-protein interactions. He also cut off the C-terminal domain, but included the HRDC. He made another construct where he deleted the dark blue and the yellow regions, but left the nuclear localization signal intact. He made another guy that's sort of the dark and the light blue, and then another little guy that's a 40, it's basically 40 amino acids of this blue region. And he's asking, okay, how do these guys behave? And so the first thing that struck us when he started looking at these constructs is that they lose the capacity to recognize DNA ends. And so this is GFP BLM, again, nicely lined up at our DNA ends. I think this is the same slide I showed you at the beginning. This is now our C-terminal deletion construct. It binds DNA just fine, but what's happening is it's going everywhere. So it's not simply going to ends, it's going all over the place. And this is true for every single C-terminal domain truncation construct he looked at, even the smallest guy. This is what we call delta 1291 to 1330. It simply goes all over the DNA. So we're not losing the capacity to bind DNA, we're losing target specificity. All right. Now, he also tested these guys in terms of bulk ATP hydrolysis activity. I'm going to point out when we chop off the whole C-terminal domain, you actually end up with a better ATPase. This is fun. These are all the other the constructs compared to wild or full-length bloom. This is ATP hydrolyzed per time. And he measured the velocity on DNA. And the fun thing here is this protein, even though it's going much, much faster in terms of turning over ATP, it's going much, much slower in terms of its movement on the DNA. And so what we're doing here with this mutation or this truncation is decoupling ATP hydrolysis from the protein's motor activity. And in this case, it's about 64% slower. I'm going to point out this protein actually was characterized in bulk biochemical assays a number of years ago by 2 different labs. And both of them showed that it had decreased helicase activity, but increased ATPase activity. So we think our result is fully consistent with the literature. And what we're adding to this now is showing that, yes, indeed, it's actually going slower. So you're decoupling motor from ATP hydrolysis. Now, sorry, I need a little drink there. Now, everything I've showed you up to this point has been work by Chow Yu. What I'm going to start talking about are some experiments that were done in Patrick's lab, this first of which was done by his longtime associate, Young-Ho Kwan, who's just been amazing to work with over the years. And so what Young-Ho did was some simple bulk biochemical assays with our 2 mutants. And what I mean by 2 is we've settled on these 2 guys as the most interesting. Our N-terminal deletion construct, which no longer generates DNA loops, and our C-terminal protein where we deleted this short track of amino acids, it's 40 amino acids roughly, and it no longer recognizes DNA ends very well. And so this is a simple measurement of DNA unwinding, taking a 2.5 kb fragment and asking, okay, how well do you unwind? And what we see is that this N-terminal guy actually unwinds it better, which is consistent with its more rapid velocity. And this guy also unwinds it. In fact, it's comparable to wild type. It's not so bad. And we think, even though it's not recognizing ends, when you have a short substrate in a test tube, that doesn't really matter. It's enough to get the job done. And so it still can unwind the substrate. You're simply putting enough enzyme on the DNA that it's not a problem. Now, we then asked, okay, what about processing when we add DNA2 to these same reactions? And what we see here is interesting in that our N-terminal deletion, which is a better helicase, actually shows processing levels that are comparable to wild type. And we think what this is telling us is something about DNA2. It's likely that DNA2, DNA degradation, this is the nuclease, remember, is likely rate limiting these reactions. But now our C-terminal deletion construct is not doing such a good job. And again, we think that that's because it's not getting to the ends very well. And these properties in general are scaling with what we're seeing in our single molecule reactions. And so what we now wanted to do was some fun experiments and ask, okay, what happens when we put these mutants into cells? And again, I'm talking about the N-terminal and the C-terminal truncation. And so this is all work done by Samir now, where he's taking these U2OS cells and knocking down either Bloom or XO1, or in every experiment I'm going to show you, we're knocking down both. And I'm going to point out, I should have mentioned this at the beginning, the reason we're knocking XO1 down is that it offers an alternative pathway to end processing in the event that Bloom is not there. So we want to get rid of both of these when we're looking at our mutants. So we're knocking these both down, and then we're putting in either full-length Bloom, a vector control, or our, in this case, C and N-terminal constructs, okay? So now, we're then asking if we take these cells and simply treat them with agents that are going to cause problems are going to cause DNA damage, either olaparib or mitomycin C, how do the cells do? And so this is our vector-only control at the beginning. The cells don't survive very well. If we put in full-length Bloom, we now get an uptick in survival in the case of both olaparib and mitomycin C. Whereas our N-terminal and our C-terminal constructs, deletion constructs, show poor survival. And so we now want to ask, why is that? Why are these guys not surviving very well? And again, I remind you, this is the one that doesn't do looping. This is the one that's not targeted to ends. It's probably pretty easy to imagine why this one's not doing so well. Okay, so now what Samir did next was to ask, okay, what happens with these guys when we treat these cells with ionizing radiation? Can we see foci forming, protein foci, both for BLM, RPA, and also for Rad51? And so we're using Rad51 as an indicator that homologous recombination can take place because we're making a processed end where we can assemble now our recombinase filament. And what I'm going to show you is we can see BLM foci in all these guys. These foci in terms of the N-terminal deletion look a little bit big. It's not unusual. We're overexpressing a GFP tag protein. In all cases, we're seeing RPA, at least to some extent. And then Rad51, we're seeing it, and it varies quite a bit. And I'm going to show you quantitation of this data now. So first thing we looked at was 53BP1, which we're taking as an early indicator of DNA damage. And although there are some differences from cell line to cell line, background to background, pretty much they're all making DNA double-strand breaks. And so we know that we're inducing DNA damage when we expose these cells to ionizing radiation. Now, all cells also show evidence of GFP BLM foci. I showed you that in the image. And of course, there are some differences from one construct to the next, but they're still forming foci. And it's interesting to think about this guy, which is not targeted to DNA ends, but it's still forming foci consistent with it going to where the DNA damage is. And I'll bring that up again a little bit later at the end. Now, what we wanted to do then was look at RPA foci. And so RPA foci are an indication that end processing is actually taking place. Now, this is our vector-only control. This is when we put in full-length BLM. And so going from here to here, we're starting to see more RPA foci that says, okay, we are generating processed breaks. We see a similar increase when we delete the N-terminus of BLM. We still see RPA foci forming. And so that's telling us that the protein is processing the DNA ends. But if we take our C-terminal construct, now we don't see that increase in RPA foci. And so we think that this guy is no longer making RPA foci because it's no longer supporting RPA foci formation because it's not processing the DNA ends. And I'm just going to point out that we also have physical assays where we're doing PCR near the break to detect the production of single-stranded DNA. And it also says that this guy is not generating single-stranded DNA, whereas these guys are. Okay, so now if we look at Rad51 foci, what we see is that with BLM, we do get an uptick compared to vector-only control, but now neither of our deletion constructs are showing evidence of strong upticks in Rad51 foci formation. And so we think that the C-terminal guy is not making Rad51 foci because the ends aren't getting resected, so there's no RPA or Rad51 loading. Whereas this one, the N-terminal construct, we do think it's resecting the ends. We do see RPA foci, but it seems to be specifically defective in Rad51 filament formation or foci formation. And so we think that there's some kind of defect associated with this guy that doesn't let Rad51 now bind to the DNA. Now, I'm going to go through two assays relatively quick and then get to the conclusions. In these two assays, what we're doing are taking GFP constructs where we have a GFP gene that's inactive and one that's active. And then we're using DNA break induced by a restriction in the nuclease ISCE1 to induce repair that results in a repair of the GFP gene so that it goes from not fluorescent to now fluorescent, okay? And we can tune these, or I should say, the assays we've adapted can attune these to detect either homologous recombination or what's called synthesis-dependent strand annealing. Sorry, SSA, I should say that. Okay, so let me walk you through this data really quick. So these are our control cells where we do nothing. This is a cell where we break the DNA and we have an siRNA that's not targeted against anything, and we can see homologous recombination taking place. This is where we cut the DNA with ISC1, but we inhibit BRCA2. And so we'd see a downtick in HR, homologous recombination. This is where we knock out BLM and XO1. We also don't see very good recombination. This is our vector-only control. This is where we put back full-length BLM. Okay, and so we do get a partial rescue of homologous recombination as long as we have full length. Now, neither of these guys are supporting homologous recombination in these cells compared to our vector-only control. And so that's telling us that, consistent with our prior results, there's some sort of defect associated with repair. Now, if we look at single-strand annealing, which is not Rad51 dependent, okay, I should emphasize that point. What we see is when we knock out BRCA2, we get an increase in single-strand annealing because we're taking things that would normally be prepared by HR and channeling them into this new pathway that's Rad51 independent. Now, if we look at our vector-only control, this is what we get. Adding back Bloom doesn't really have much of an effect. Our C-terminal deletion also doesn't have an effect. But adding this N-terminal deletion, now we get a substantial increase in single-strand annealing. And we think that's because this guy is making single-stranded ends that are loaded by RPA, but they're not loaded with Rad51. And so Rad51 actually inhibits this pathway. And so we think that that is fully consistent with this idea that somehow this N-terminal deletion construct is deficient for Rad51 filament formation. All right. So now I'm going to go to my summary slide. And I hope that I've convinced you that end resection in eukaryotes is somehow coupled to the formation of this large single-stranded loop, and that the N-terminal domain of BLM, Bloom helicase, is somehow contributing to this formation. And cells expressing this mutant are deficient for DNA repair in the presence of DNA damaging drugs. And it seems as though this mutant can support end resection in vivo, but Rad51 is failing to form on these ends. And so our hypothesis then going forward is that there may be some kind of link between loop formation during end resection and the assembly of the Rad51 presynaptic complex. I've colored this blue and added a question mark because this is, of course, speculative at this point. And we're curious, what is driving these looping interactions? Is it BLM interactions with single-stranded DNA or perhaps BLM interactions with RPA? We don't know. And we're also curious whether BLM itself may be somehow loaded or involved in loading Rad51 onto the DNA ends during resection. And of course, we always have the question of, can we separate or generate mutants that separate looping from Rad51 loading? We don't know the answer to that yet. Okay, and I also want to make a point here. The key bacterial system, so for example, REC-BCD and E. coli, it also makes DNA loops during end resection, but we don't know why. And so it seems as though, and I want to make this point clear, REC-BCD, which processes DNA ends in E. coli, is completely different from BLM DNA2 RPA, which is involved in processing in eukaryotes. But still, both systems are forming loops, and we're wondering, is that a coincidence, or is there something to loop formation that's important for recombination? And we don't know the answer to that. Now, in terms of our C-terminal domain, we know that now the HDRC motif helps couple ATP hydrolysis to translocation. We have no idea how that takes place. It's interesting because within the crystal structure, this domain seems to be well away from any DNA binding motifs, although you can always think about new structures that might change that. We've identified a new peptide within BLM. It's flanking the HDRC, but it's not within the HDRC itself, that seems to be necessary for end resection, even though it's not messing up any of the other properties, either DNA binding, ATP hydrolysis, or helicase activity. It's specifically affecting recognition. And we can show that this mutant is recruited to sites of DNA repair, but that it's not actually targeted to DNA ends, and it seems to be severely compromised for end processing, both for formation of RPA and Rad51 foci, and it doesn't do HR. And so one thing that we're thinking that this is telling us is that because it's going to foci, actual recruitment to the repair foci may be a separate process from then direct recruitment to the actual DNA ends. And again, that's sort of a speculative thinking that we have to sort of work out in the future. And so I'm happy to end there and address any questions that anyone might have. Not entirely sure how this is going to work, but I think Susan's going to read your questions to me. So we'll see how that goes. And I'll just thank everybody for listening to me.
Thank you, Eric. I'm sure if we could see people, there would be a lot of hands clapping right now. I'm not sure entirely how this works either, but I think we'll work it out. It was a beautiful talk, and there are some questions in the chat. Please continue, everybody, to add your questions, and I will go through as many as we can. So the first one is from Rachel Johnston, and it's more of a technical setup question about controls for your curtain assay. She asks, you noted that the BLM localized to the DNA ends using GFP labeling. Did you have a control for localization for a protein which would localize to another location on the DNA? So rather than it being a buffer flow issue, it is true localization.
Yes. So yeah, over the years we've published, I don't know, 100 papers, north of 100 papers now. And in several of those, we've published proteins that bind to specific sites within the DNA. I think one of the more fun ones that we did was with Cas9, actually, and we could target it basically wherever we wanted within the DNA just by changing the guide RNA. We've also done studies. I should have mentioned this. This is lambda phage DNA that we're using. It's nothing fancy. We can buy it. Nice thing about lambda phage is it has RNA polymerase promoters, and we know we can put E. coli RNA polymerase in there, and it goes to all the right promoters. We can put in also let me think about this, a nuclease deficient version of EcoR1. It goes right to the EcoR1 sites. And we can put in a mismatch in that DNA, and we can target the mismatch recognition proteins, MSH26, right to the mismatch and not to the end of the DNA. So we don't think flow is is causing it to go to the end of DNA. And I should also note, we're often asked, well, how do you know force is not influencing this? And I can't say with certainty that it's not, but what I can say is that we're not exerting a tremendous amount of force on these molecules. This is one of the reasons they sort of look like they're flipping back and forth a little bit. We're sort of down in the sub single picomolar digits, piconewton digits, just enough to extend the DNA about 70% of its full contour length. So we don't think we're doing that. Now, I will note we have seen examples of enzymes where we push it off the DNA, at least we think we do, but I don't think that's taking place in this example.
That's great, thank you. So Tom Snyder has a couple of questions that I'll ask on his behalf. The first is, did you get the number of ATP hydrolyzed per step in wild type BLM?
Tom, I love that question. I hate it too. This is of course he must be a motor protein aficionado. This is a question that often comes up, and that is, how much ATP do you hydrolyze per step? And I always make the simplest assumption, and I think this is consistent with structural data for all helicases that I know of, or most of them anyway, that you're taking a single nucleotide step. And so it's of course of a great interest to know, okay, is one step correspond to one ATP? And Tom, I would love to be able to answer that. I can't. We haven't made that measurement yet. It's not such an easy measurement to make correctly. And really the only way that we can do this is to essentially do bulk biochemical measurements where we're measuring ATP hydrolysis per unit time and then compare it to the velocity. Now, the challenge with that in this particular system is that we know that BLM binds to ends. We also know that it binds internal positions. And the velocity that it moves in those two different configurations is very different. It moves much faster when it's bound to an internal site. So any bulk biochemical ATP hydrolysis data is going to be a mixture of things that are bound to ends and things that are bound to internal sites that we don't have a good way of deconvolving that. But I do appreciate that question, Tom, and I would love to know the answer myself. So,
yeah, that's a good point. The bulk biochemistry is not going to really answer that well, I don't think either. He also asks, have you looked at the loop using cryo-EM? Maybe it is a coiled helical structure.
It could. We have not done that yet. We're really. I'll just say it. In a lot of the projects that we're working on, we've kind of come into these barriers where it's like, man, I would really like to have a structure of that. This is not just this project, but of course it applies here as well. And so we really need to start doing some cryo-EM on some of the things that we're looking at, and try to get some structural insight. That, again, another great question, which I unfortunately can't answer very well right now. Sorry, Tom.
You have a lot of colleagues at Columbia that could help with that in the cryo-EM.
Yeah, they're all trying to solve a virus structure, antibody structures now.
Okay, next question. Michael,
we're going to do it ourselves.
Yeah, good for you, and you can. Asks, it is also interesting to note that a Rec-A loading function has been suggested for Rec-B-CD.
Yeah, so that's Michael Lichten. Is that what you said, Michael? Man, I wish I could see you and hear you. This is one of the things I don't like so much about Zoom. That's a great point, Michael. It's very clear that Rec-B is somehow involved in loading Rec-A, and we also know that BLM interacts with Rec-A, or sorry, not Rec-A, Rad51. See, I'm getting too excited. And so we would love to be able to see this, to see coupled Rad51 loading in our system. We're not there. We're going to move in that direction. The challenge, Michael, is that, and you're probably well aware of this in a test tube, because it's an artificial system, an important caveat that you have to contend with is that Rad51 doesn't distinguish very well between double and single stranded DNA. And so I know that if we just throw Rad51 in there willy-nilly, it's going to go everywhere. It's going to get in the front of this thing. It's going to get behind it. It's going to cause all kinds of chaos. I suspect we can overcome that by perhaps one or two ways. The first of which is to perform the reactions under conditions in which Rad51 loading itself on its own is compromised, meaning a lower concentration of Rad51 that is necessary to get on the DNA by itself under the assumption that BLM might help it get on there. And so we're definitely thinking about that. And then the other way, as you know, there's a number of different proteins that are involved in helping to get Rad51 on DNA. So mediator complexes, you know, like BRCA2, for example, or Rad51 paralogs, possibly. We would love to be able to see coupled reactions in where we're doing this processing, we're putting in the mediators and we're putting in Rad51 and try to see where does everyone fall in place. We're not there yet. Sorry, Michael.
Okay, we're going to take one final question, but before that, let me repeat the CME code for today. For those of you who need it, it's 37930. So I thought the last question should come from a fellow LMB person. Jinwei Zhang is one of our newer investigators at LMB, and he asks, fantastic, he says, fantastic talk, Eric. Do you think that this single-stranded DNA forms a giant single loop or many small loops due to topological constraints? Could it be forming numerous DNA hairpins as a long single-strand RNA loop does?
Yeah, so that's a great question, Jingwei. I don't know if we've met, so you know, I hope I'm not forgetting you, but if not, I'm sorry that I couldn't come there and meet you. So those are two different things, kind of. So exactly as you say, you could be forming multiple sort of hairpins that are not involved in a protein-protein interaction, but are involved in a sort of DNA-DNA pairing interaction. Or you could form multiple contacts, perhaps, with the resection machinery. I don't think that either of those is taking place. And the reason I don't think so is that when we see, there's a couple of things. First of all, when we see the loops break and that DNA gets long again, it tends to happen all in one step. And so that tells me that we've ruptured a single contact. Of course, I can't say that maybe there were multiple contacts and it's the last one that you see. That's also a possibility. I don't think hairpins are forming, at least not to an excessive amount. And the reason for that is that we know RPA, it's one of its primary roles in these reactions, or in reactions like this, to prevent hairpin formation. And so I'm sure if we didn't have RPA there, that whole thing would be knotted up like crazy, just like you would envision an RNA molecule. But when we have RPA there, I think it's preventing that from taking place. So hopefully I answered your question, Jinwei, and hopefully I can meet you someday too.
We all hope that. Yes, that would be really nice. So I think it's probably up to me to thank Eric again for a lovely talk. I had questions too, but we couldn't get to everything. And we have a full trainee schedule for T, which was supposed to give Eric a little bit of a break, but there's not much of a break left.
Yeah.
This is a gorgeous system. It's so elegant. There's so much you can do with it. It's going to be very exciting to see where this goes from here. So on behalf of all of us at NIH, we miss you and thank you so much for giving this talk today.
Thank you, Susan.